Hello students, today we will learn about genetic engineering of plants and its methodology. Let's begin. By the conventional plant breeding techniques, significant achievements have been made in the improvement of several food crops. These age-old classical methods involving gene transfer through sexual and vegetative propagation take very long time. For instance, about 6 to 8 years may be required to develop a new rice or a wheat by sexual propagation. Rapid advances in gene structure and function, coupled with the recent developments made in the genetic engineering techniques, have dramatically improved the plant breeding methods to yield the desired results in a short period. Plant genetic transformation technology basically deals with the transfer of desired gene from one plant species to another, with subsequent integration and expression of the foreign genes in the host genome. The term transgene is used to represent the transferred gene, and the genetic information in plants is broadly referred to as plant transgenesis. The genetically transferred new plants are regarded as transgenic plants. The development of transgenic plants is the outcome of an integrated application of recombinant DNA, that is rDNA technology, gene transfer methods, and tissue culture techniques. Now let's know about the why transgenic plants we use. What is the benefit? All details we will discuss. It is only through the recombinant approach, that means genetic engineering of biotechnology, new genes with desired characters can be introduced into the plants. Further, it is possible to manipulate the existing genes to make the proteins with suitable alterations. Example, increase in the content of an essential amino acids. The most important regions for the developing transgenic plants are now we will discuss. To improve, first point is to improve agricultural, horticultural and ornamental value of plants. And, second point is to develop plant bioreactors for inexpensive manufacture of commercially important products, example proteins, medicines, pharmaceutical compounds. To study the action of genes in plants during development and various biological processes. Now let's know about the genetic traits introduced into transgenic plants. Since plant cells are totipotent, that means a single plant cell can regenerate into a whole plant. The genetically engineered cells with new genes can produce a transgenic plant. This plant carrying the desired trait will give rise to successive generations. Many genetic traits have been introduced into plant through genetic engineering. First point is resistant to herbicides. Second is protection against viral infections. Third is insecticidal activity. Fourth is improved nutritional quality. Fifth is altered flower pigmentation. Sixth is tolerance to environmental stresses. Seventh point is self-incompatibility. Now let's know the criteria for commercial use of genetically transferred plants. For large-scale commercial application of genetically engineered plants, there are some requirements. Let's know about the requirements. What are the requirements? First requirement is introduction of desirable genes to all plant cells. Second part is expression of cloned genes in the appropriate cells at the right time. Third point is stable maintenance of new gene inserted. Fourth point is transmission of new genetic information to subsequent generations. Now let's know about the Gene transfer method. What is the gene transfer method? Gene transfer technique in plant genetic information are broadly grouped into two categories. First is vector mediated gene transfer. Second point is direct or vectorless DNA transfer. Now let's know first point. First method is vector mediated gene transfer. Let's know about this. Vector mediated gene transfer is carried out either by agrobacterium mediated transformation or by use of plant viruses as vectors. First is agrobacterium mediated gene transfer. What do we mean by this? Agrobacterium tumefaciens. Agrobacterium tumefaciens is a soil-borne gram-negative bacterium. It is rod-shaped and motile and belongs to the bacterial family of Rhizobiaceae. Agrobacterium tumefaciens, or in short A. tumefaciens, is a phytopathogen and is treated as the nature's most effective plant genetic engineer. Some workers consider this bacterium as the natural expert of inter-kingdom gene transfer. In fact, the major credit for the development of plant transformation technique goes to the natural unique capability of Agrobacterium tumefaciens. Thus, this bacterium is the most beloved by plant biotechnologists. There are mainly two species of Agrobacterium tumefaciens. First is Agrobacterium tumefaciens that induces crown gall diseases. Second is Agrobacterium rhizogenes that induces hairy root diseases. Now let's know about crown gall disease and type plasmid. Almost 100 years ago, two scientists named Smith and Townsend postulated that a bacterium was the causative agent of crown gall tumors, although its importance was recognized much later. As A. tumefaciens infects wounded or damaged plant tissues, induces the formation of a plant tumor called crown gall. The entry of the bacterium into the plant tissue is facilitated by the release of certain phenolic compounds, that's acetosyringone, hydroxyacetosyringone, by the wounded sites. Crown gall formation occurs when the bacterium releases its diplasmid, tumor-induced plasmid. Diplasmid means tumor-induced plasmid into the plant cell cytoplasm. A fragment of diplasmid, referred to as tDNA, is actually transferred from the bacterium into the host, where it gets integrated into the plant cell chromosome, that is, host genome. Thus, crown gall disease is a naturally evolved genetic engineering process. The tDNA carries genes that code for proteins involved in the biosynthesis of growth hormone, that is, auxin and cytokinin, and novel plant metabolites, namely opin, that means amino acid derivatives, and agropines is sugar derivative. The growth hormones cause plant cells to proliferate and from the gall, while opins and agropines are utilized by Agrobacterium tumefaciens as sources of carbon and energy. As such, opins and agropines are not normally part of the plant metabolism. Thus, Agrobacterium tumefaciens genetically transforms plant cells and creates a biosynthetic machinery to produce nutrients for its own use. As the bacteria multiply and continue infection, crown gall develops, which is a visible mass of the accumulated plant bacteria and plant material. Crown gall formation is the consequence of the transfer, integration, and expression of genes of tDNA of Agrobacterium tumefaciens in the infected plant. The genetic transformation leads to the formation of crown gall tumors which interfere with the normal growth of the plant. Several dicotyledonous plants are affected by crown gall disease, example grapes, rose, stone fruit trees. Now let's know about organization of Thai plasmid. The Thai plasmid exists as independent replicating circular DNA molecules within the agrobacterium cells. The tDNA, that means transfer DNA, is variable in length in the range of 12 to 24 kilobase per or kb, which depends on the bacterial strain from which